Well, hello there and welcome to Coffee Chats with Experts. I'm your host, Laura Moriarty, and I'm really excited today to be leading the panelists in a discussion around mixed mode chromatography. All right, so who do we have joining us? Good morning, gentlemen.
Good morning.
Hello.
Morning.
All righty, so let's get started. So first up, we have Dan. Hey, Dan.
Hello. Well, good morning for coffee.
Morning. So Dan is based here in California as well as myself, and he has 20 years experience in chromatography across a variety of settings. And he's been at Bio-Rad for about 15 years now. And he's been connecting life science researchers with all kinds of tools and solutions to expand their chromatography expertise for biomolecule purification. So welcome, Dan.
Yes, thank you. Thanks for having us.
And next up is Bill. Hey, Bill.
Hey, how's it going?
It's all good. So Bill's over there on the east coast of the US, based up in Pennsylvania, and he's been doing downstream process development for about 23 years now. And although he's only been at Bio-Rad for 5 years, he has managed to make a great impression and support a lot of customers on the process development side of things. So welcome, Bill.
Thanks, Laura.
Alrighty, and last but not least is Mark. Hey, Mark.
Hey, Laura. How's Brexit working out for you?
Well, I have my British mug with me this morning, so you know, we'll get to that if questions allow us to. How about that? So Mark is also based here in California, and he's been at Bio-Rad for 15 years now. And prior to that, he spent 30 years in industry doing process development. So I think, you know, as I like to say, I think we have the right people on the call today. And so without any further ado, I will shift us over to a couple of housekeeping items for those in the audience. And so, lovely audience, we love to get your questions. So please do enter them in the Q&A, and we will get our panelists to give you their tips and tricks. And if you're listening on demand, you can still enter your question, and the panelists will get back to you via the email address that you've used to register for the event. All right, so I think that is it. I think we are good to go. So I always like to kick us off with a couple of just kind of warm-up questions. And so panelists, are you ready?
Ready to go.
Yep.
Ready? All right, here we go. So the first question up is, what defines a resin as a mixed mode resin? So great introductory question that's easier to spell than say. Who would like to kick us off? Dan, go for it.
All right, and then Mark and Bill, being the experts that they are, I'm sure they'll have follow-on comments to make. So first of all, it's called mixed mode resins. Mark, because it has multiple modes of interaction with the molecule that's to be purified. So if we start back with traditional resins, those are the single mode, like ion exchange Q, ion exchange S, DEAE maybe, and also HIC, hydrophobic interaction. So those are all single mode interaction resins with the molecule. Mixed modes will combine those two into one single ligand, into one resin. So for example, Nuvia C prime from Bio-Rad, Nuvia A prime 4A from Bio-Rad. You also have from Cytiva, Capto Adhere, Capto MMC. Those will incorporate weak cation exchanges or strong anion exchanges with hydrophobicity on the ligand. Also, Bio-Rad has another one called ceramic hydroxyapatite, or CHT for short. And that's also mixed mode. It's a little bit different type of resin itself, but we can get into that a little bit later on. But CHT is calcium affinity and cation exchange resin. So we'll start with that as a mixed mode. So mixed mode is multiple modes of interaction with the biomolecule.
And I'll just add one thing while I'm thinking about it, which is that while today most people think of mixed mode as the interactions are occurring on the ligand, it's also possible to have mixed mode interactions between the ligand and the matrix of the resin itself. And I mention that because, for example, our macroprep line of resins, we have all 4 flavors of ion exchange, but the macroprep backbone is rather hydrophobic. So there is actually, those actually are also functioning as mixed mode resin. So,
oh, interesting. Anyway. I got a little extra education there this morning as well. I didn't know that. Cool. That one will come in handy for the that one will come in handy for the pub quiz on Friday night. All right, so that was a great warm-up.
Did you say pop quiz or pub quiz?
Pub quiz, come on. Going with the British theme, right? Pub quiz. Okay. All right, so here's our next question. Is there a generic strategy that you could recommend for screening and optimizing your method with a mixed mode resin? This seems like it could be a long question, but let's try and keep it short so we can get to some others. And Bill, I see you have your hand raised.
Sure. Yeah, we definitely have a generic strategy within Bio-Rad that we recommend. Initially, we look to find optimal binding and elution conditions using strictly pH. When we've actually done that with customers, they've been shocked that you can actually get a good process without the addition of any salt. But if you're not able to get good resolution, then we start looking at introducing salt. And finally, if that doesn't work, then there's some other modifiers that you can add in there, to further enhance your separations.
All right, brilliant. So that should give people a good place to start. All right, so I'm going to get us over to some audience questions now. Here we go. All right, this is a very polite question. I like this one. Sir. Sir, I love it. So I might do it with a kind of British style then. Sir, how can one improve protein purification, especially when one is doing a crude extract from animal tissue? There you go. That's a British spin on the question. Who would like this question?
So I'll take it. And the true British version of this question would be, sir, could I please have some more? Because that's essentially what this question is asking about, right? So the answer is you really have to look at what the purpose of the initial steps are. Depending upon what you're looking for, the first thing I generally suggest, if you're not getting the separation you want, is you could try a variety of mobile phase modifiers, and each step has mobile phase modifiers that you can add, and some work better than others. So I know on mixed mode, for example, as Bill indicated, adding things like arginine, for example, can dramatically alter the behavior of proteins on the resins. If you're working with CHT, which is insensitive to surfactants, you can think about adding all sorts of different surfactants as a wash step. So generally, you want to think of things like that to begin with.
Brilliant. So some good tips there. Thanks, Mark. Anyone else want to throw in a couple more ideas there to that one?
Yeah, I just agree with Mark.
Laura, go ahead, Bill.
Sorry, Mark.
No, go ahead.
I mean, a lot of it really depends on, what you're trying to purify. A lot of times, when we have conversations, we want to find out, about their target molecules so that we can then offer better suggestions for which resins to really target for the purification.
Yeah, let me add one other thing, and I say this every time, and so does Bill, every time we go out and talk. If you ever have any questions about our products, either you don't know how to use the best way, or you are and they need to be better, or anything, please let us know. I mean, this is my job, or a good part of my job. It's also a good part of Bill's job and Dan's job as well. If you don't call us, we can't help. But if you call us, we're always happy to talk with our customers and try to suggest things that we have past experience with that can help in a particular situation. So.
Mm-hmm. Absolutely, and I can vouch for these three gentlemen. They are very well versed in chromatography, mixed mode chromatography, protein purification, process scale purification, and all that jazz. Okay, I have another question coming in from the audience, so I'm going to keep us moving. So this is a long one. Okay, so considering there are multiple modes of interaction, how can we pick the right resin for the protein of interest, especially in research when we do not know a lot about the protein other than maybe a theoretical PI or molecular weight? We usually have less knowledge about hydrophobicity. Who would like to tackle this? Bill.
I think this actually dovetails off of the initial question you had with me about the generic screenings. So usually when we talk to customers, PI is probably the first thing we ask for because that will kind of dictate which resin you're going to be looking at. I mean, if you're looking at a low PI, if you want to bind, then you can bind it with some higher pH. So definitely once we have this minimal amount of information, we can definitely help you determine which mode and interactions to try with. And like I said, start initially with, just a pH elution. That doesn't work, then start adding in salt and then modifiers.
Great. Anyone catch it? Dan? Yeah.
Yeah, just add on to what Bill just said. There's a small, in 96 well plate formats, small format, there's also a screening procedure called DOE. Most folks who have done work with mixed mode are familiar with that, but design of experiment, and that takes into different concentrations, different angles of salt and pH, for example, and that's a really good first step to try to get a region of binding or flow through. It can either make it bind or not bind, so it's one or the other. And then you can also do a little DOE at the end of it too to figure out how to do the elution. And that's so speaking to the modifiers that Mark had mentioned as well. So just to add a little to what Bill just said.
Yeah, I'll add one thing on that, which is a slightly different answer is that, I mean, in some cases, so Bill is certainly right. Given the PI, it gives you a general first cut of where to start, just like with ion exchange resins, right? I mean, it's the same thing. However, if you pick the right resin, you can't. I mean, the simple answer is you really have to try them all, especially with mixed modes, where one cation exchange hydrophobic mixed mode does not perform the same as some other manufacturers' cation exchange hydrophobic mixed mode because the ligands are different. Steve Kramer has published a lot on this, and good news, bad news for people at the bench. You just have to try them all and see what happens. And Dan is right. We at Bio-Rad, we have both 96 well and 1 and 5 mil or 200 and 600 microliter robocolumn style columns that you could easily test in a robotic system. So,
and if you don't have a robotic system, we do have the mini spin columns that we do DOEs with as well, where you add your own resin. So I've had customers say, "Oh, do we have to use Bio-Rad resin?" I'm like, "No, you can use anybody's resin." But
shh.
Well, hi, I'm being honest.
I know. We're in it for the science, right? It's good.
Yep.
All right. So yeah, mixed mode resins, just by virtue of how they're designed, right? It's not just one flavor. You've got multiple flavors, multiple interactions going on. So it does make sense that it might be a little bit more upfront work at the beginning, but certainly the benefits outweigh that initial effort, right, gentlemen, with the fact that you get, some really unique selectivity with these resins. Dan?
Yeah, so just to add, sounded like the question, if I remember the question correctly, at the very end of it, it was something about the different if you don't know the hydrophobic character of the molecule itself, right? And so in answer to that would be that there are, of the resins that are on the market between Bio-Rad and Cytiva, formerly GE, the ligands will actually have different levels of hydrophobic character as well. And that can be measured if you have the right equipment as well. But without having actually measured, just as Mark had mentioned, you'll just have to try different resins because all the different resins, the point being is that different resins do have different levels of hydrophobic character. And that could make a difference in your selectivity, ultimately.
Brilliant. All right, so I'm going to move us along to our next question. All right, could you do empty full AAV virus separation using mixed mode resins? Ooh, I love this one. Very timely with everything that's going on in vaccine development. Who would like this one?
So the answer is yes. The answer is yes, and Bill actually has a lot more details on that, so I'm going to let him talk about our results with at least one of our mixed mode resins. See how I did that, Bill?
I like that. I like that. He took us there, and then he's like, "No, you can have it."
I'm like, wait, can I hear this here? A bus?
Yeah.
Yeah. So with we have the AD5 purification, and in that study, C prime is used as the capture. And if you look at the main elution peak of the optimized process, there's actually a double peak. Now that study, when it was done, it wasn't looking at empty versus full, but it's quite possible that that 2-peak elution that you're seeing could actually be the separation of the empty versus full. So it is definitely capable of being possible with, both of our resins.
Yeah, I know that that's certainly a pain point for many folks out there right now. So that's a good piece of data to share. Okie dokie, thank you. I am going to keep us going. Oh, here's a good one. All right, so this person has a 5-step purification process, flipping neck. What do you recommend to reduce the number of steps? Oof. Who would like this one?
Bill, you want to start that? I can go after you.
Bill again.
No, sorry, Dan. I didn't hear you actually say. I heard a name, but not who.
Yeah, this is a common question that comes up, and we enjoy this question. So I'll start it, and then Bill and Mark can add on. But so we essentially can use a mixed mode resin to go. In this case, if it's a 5-stepper, we can reduce it to 4. Right, so it'll take a 2-step into 1, and that's as Mark was saying earlier. There's the ion exchange component, let's say, from the mixed mode resins, and you got the hydrophobic. So if you just treat those two individually, and if your procedure has a back-to-back Q to a HIC, that would be a strong anion to a hydrophobic, like a Nuvia A prime or a Capto adhere. Q is usually used to get rid of DNA, and then HIC is usually at the end as a polish step. And so you can eliminate 1 step by combining those 2 into 1. And we have a customer who actually has been doing that. Several customers actually looking into that. In which case, in that case, the real scenario was a 3-step into a 2, where they had a protein A capture followed by a Q, and then a HIC. And so they wanted to reduce 1, and the reason for that is to increase your process economics or improve your process economics. Save time and save material buffers, so whatnot. So that's how mixed mode can be used to reduce the number of steps.
Yeah, Bill.
I would add that I think it's possible with CHT because it does remove so many different impurities that depending on what each step is doing in a process, you could maybe even eliminate more than 1 column step, because CHT, I always like to call it like a giant sponge, because it just seems to remove so many different impurities that, I mean, you could potentially, knock out 2 steps with go from a 5 to a 3 with CHT even.
Good point.
Oh, has CHT been used in virus or viral vector purification? So who would like this one?
I guess I'll take it.
Okay.
So the answer is yes. The answer is yes, and it's been used to purify many different viruses with very high yields. A lot of viruses tend to bind very, very tightly to CHT, and so the separation between viruses and a lot of the host cell impurities tends to be really good, and the yield also tends to be really high. So yeah, the answer is yes, and we can certainly supply you with more information on that.
Yeah, definitely a timely question there. Thanks, Mark. Are you all plugged in now?
I'm plugged in.
Brilliant. All right. Okay, next up from the audience, do you recommend certain mixed mode resins for capture step or polishing step? Oh, okay. So where in the process should somebody use a mixed mode resin? Who would like this one? Bill.
I mean, I think with the 3 that we're talking about with Bio-Rad, A prime and C prime, both can easily be used as capture or polishing steps, depending on, the product that you're doing. CHT can be a little challenging as a capture step because, as I mentioned before, because it does bind so many things. Sometimes you don't necessarily want to have crude feedstock. Now, if you have a pretty pure feedstock coming off of out of culture, you could use CHT. We do actually have some applications where it has been used in a single step purification with CHT, but most people use CHT more as a polishing compared to a capture step.
Brilliant. Dan, yeah.
Yeah. So, and there is a tech note, an application note that will be coming out here, but we have a customer who's used it to capture recombinant protein directly from feed. And so the benefit or the strength of mixed mode resins is that it is salt tolerant in the sense that you don't have to, it'll bind your molecules at whatever the salt that's already in the feed. So you don't have to dilute it. You don't have to do any filtration. You don't have to do any diafiltration to dilute it out. And so in this case, they loaded E. coli feed directly onto A prime, Bio-Rad's A prime, and used as a capture. And then CHT was used as the polishing step, and it worked really, really good. So we'd imagine with the monoclonal antibody from let's say a CHO cell, mammalian cell, it'll probably work the same. I don't have a example of that right offhand right now, but with the salt tolerance idea, it should work as well.
Oh, that's great. Thank you. All righty. Let's get another one from the audience here. Okay, so considering uncertainties come with mixed mode, can we consider them as a first line of selection or after traditional ones? Good question. Who would like this one?
So I'll take that. I think, I mean, it depends upon what you're trying to achieve, right? So if you have a very dirty feed stream with a low concentration protein, then mixed modes are not what you want to start with. You would want to start with something like a traditional ion exchanger where, as everyone knows, ion exchanger capacity, in general, they're pretty much the highest of any type of resin. So I would probably start there, since water is the largest contaminant. The first thing you want to do is dewater your feed stream. Having said that, these days, given the ability to use, at least for many people, robotic systems, in some senses it doesn't matter. So if you have let's call it a typical feed stream, you could probably use them as a first line of selection. And again, it kind of depends upon what you know about your protein a little bit and what you know about the feed stream. If you know you have to dilute your protein tenfold to get it to bind to an ion exchanger, but on some of our mixed modes, you can actually load at 15 to 20 millisiemens per centimeter, which is typically where feed streams tend to be, right? Then you might want to think of using a mixed mode as the initial step, because maybe you don't want to have to use that much water to dilute out your feed stream, even if it's online dilution. So the answer is yes and no, depending upon really some of the properties of what what you have in your process stream.
Bill.
Yeah, I mean, I would agree too, Mark. I mean, a lot of people are going to go with traditional cation exchange first, and a lot of times that's kind of their go-to. But mixed modes can be a good first option. We had a customer that they had a really low expressing protein in E. coli, and the only resin that they were actually able to bind, capture it, and elute it adequately was the Nuvia A prime. And it actually took them using, it was arginine to elute. So it was pretty impressive. I mean, they looked at pretty much everything. And actually, before A prime came out, we had actually gone through our whole traditional resin library within Bio-Rad to no success. So then when A prime came along, we were able to actually find something that worked for them.
Yes, like that ice cream shop, what, Baskin Robbins with, was it 39 flavors or something? 31 flavors,
31 flavors.
Yeah,
yep.
So it's like that, right? Chromatography. Okay, here we go. So let's see what else is keeping our audience awake at night. Okay, here we go. So some people have promoted the use of CHT in a calcium mode up to 10 millimolar calcium in the buffer. What are your thoughts on this variation of running CHT? Also, other eluents such as sulfate and borate have been proposed. Any comments? So this is kind of a two-parter. Who would like this one?
So I guess I'll take that.
Go, Mark.
So yeah, I know the work you're talking about. We don't have a lot of people using what is essentially calcium saturated CHT. It certainly does perform differently, and you could try it. I'm not a huge fan, just because you have to make sure to keep the surface calcium saturated. Otherwise, you don't know what kind of interactions you have. But, you know, it's worth trying. If you're not getting the results you want and that works really well for you, you might as well. In terms of the other eluents, I really do not recommend them. We did experiments a good number of years ago where sulfate was found to be really bad for CHT longevity. The problem is you really need to keep phosphate in all of your buffers, and we have lots of information about that, and so generally I don't recommend it. That said, because borate is typically used at a high pH, I do know of at least one customer that is working in a borate system, and it seems to perform well for them. But again, you would only do that in a situation where you need a high pH.
Got it. Anyone want to add to that? Bill.
Yeah, just to add on about the calcium. I mean, I do know that we've run into scenarios where when we've done our, buffered ISR, if you don't kind of pay attention to the amount of calcium that you're adding onto the CHT, you actually can impact your separation. We've seen shifts in the chromatographic peak over time. So, there is a limit to the calcium addition and even running in that calcium mode. So that would be something that if you do want to explore that, please reach out to us and we can kind of steer you in the direction to make sure you're not coming back after 2-3 cycles saying, oh, the thing isn't working at all. What's going on? Because we've had that happen.
Yeah, there's nothing like a frustrated scientist. I get that. I've been there myself. All right. So here is another question. So what are the different modifiers apart from salt that can be added to elute proteins in mixed mode? This is a great question. Who wants this one? Oh, Bill.
No, Mark started breathing first.
All right then.
Oh, okay. So there's a whole range of them, and they each do different things. Arginine, propylene glycol, urea, surfactants. I mean, those are some of the ones that you could use, and they're what they do in solution is pretty well known. There have been lots of papers published on that, and again, I know that Steve Kramer has published some information on the effect of at least some of these mobile phase modifiers on different mixed mode resins. So I would point you to his papers on the subject. Interestingly enough, the results are not always what you would expect. So, for example, I believe it's on our Nuvia A'4A. BSA binds Nuvia A'4A at a certain binding capacity, and it's as Dan has indicated, it's an anion exchange mixed mode resin. So you would think adding propylene glycol to the loading buffer would decrease the binding capacity because it's preventing hydrophobic interactions on the resin. Turns out it's exactly the opposite. It almost doubles the binding capacity of either BSA or HSA. I forget which one. And we're not entirely sure why that is, but we think that the propylene glycol is probably modifying the BSA structure in some way so that sites which were not previously accessible to the ligands now are. So you never know exactly what you're going to get. So those at least are some of the modifiers. And again, please let us know, and we're happy to talk with you about how much you could use, which ones you could use, and so on and so on. So.
Absolutely. Sounds like it will be a good, almost therapeutic discussion to figure out which ones to go for. Anyone, anything to add on this question? No? Okay. Mark got them all. That's brilliant. Thank you, Mark. So let's, woohoo, right? You get gold stars. All right. Oh, here's a good one. So how does CH, yes?
I was just going to say regarding the mobile phase modifier, just one last piece. So there are a couple of webinars that Bio-Rad had executed just not too long ago, and they're posted on our website. And in it, all I'm trying to say is there is a slide or two that discusses these modifiers that Mark was just talking about. So just want to let you know that. That's just letting you know there's something posted on our web in terms of webinar.
Mm-hmm. Fantastic. Thank you. All right. So this next question here, this is a good one. How does CHT differ from other mixed modes? Who would like this one?
Okay. Go ahead, Dan.
Mark.
I was going to let Dan go.
So it differs. Actually, it differs in a number of significant ways from other mixed modes. One is that it is, as Dan alluded to earlier, it is calcium affinity cation exchange, right? It is the only calcium affinity mixed mode resin that's out there. So for those of you not familiar with calcium affinity, if you work with IMAC type resins, it's like that, except instead of copper or nickel or whatever, it's calcium, and that requires clusters of negatively charged groups. They don't have to be carboxyl groups. They could be phosphate groups. They could be sulfate groups, a whole variety of things. So that's one way in which it differs. The other way in which it differs is, unlike virtually all other resins which are out there, well, most other resins anyway, it's incompressible. It's a mineral. It's a particular form of calcium phosphate. So when you get ready to actually try and pack a column, please let us know. We have a really good applications guide on our website, or one that can be given to you by any of our process specialists or Bill or Dan. You need to read the instructions on how to use it, because if you try to pack it like you're packing 7X G25, you're going to be in for quite a shock. So those are at least two of the ways which it differs. I guess there's also a third, in the sense that due to the calcium affinity, there are certain classes of molecules that essentially bind very tightly and don't come off until you strip the column. And so no matter how you develop the process, you really always get good clearance of those things like DNA and endotoxin and retroviruses, which are not always guaranteed with other resins. Pretty much they're always guaranteed with CHT. And again, this is a topic that Bill and I speak on extensively. So if you're really, if you want more information, we'd be happy to set up webinars with your company, because it's about an hour or a 2-hour talk. But, in a nutshell, that's it. It also comes dry rather than in a slurry. So.
Yeah, it's almost like you could do a spot the difference between CHT and other mixed modes, right? It would be a very big spot the difference chart. Bill, you wanted to add?
Well, yeah, and just one other thing about CHT, unlike traditional resins, CHT is not a resin in that respect. I mean, it doesn't have a base bead with a ligand attached to it. CHT, the whole bead itself, the whole particle is the ligand. So it's pretty unique in terms of that. So even as you get into the life cycle, as long as you have that particle, it's going to react the same way. Whereas, traditional resin, you might start losing, capacity as, ligands maybe get off through the cleaning regimen and things like that. CHT is always going to act like CHT because it is, the ligand is the bead.
Dan.
All right. If there's anything to take away from that question, from CHT and how it differs from other mixed mode, I just wanted to reiterate back on what Bill just said. It's CHT is a mineral. It's not a polymeric resin, right? So, and then when Mark said it got into the compressible versus incompressible. So if there's anything, CHT is easy to pack. It's easy to pack. It just packs differently. So just have to, as long as you understand that difference, then you're good. But CHT, you can't bring down the adapter and have it touch the bed. It'll crack the particles. Whereas for the compressible, then as we all know, you bring it down and you compress a little bit further. There's a compression factor that's usually provided in our manuals. Usually like a 1.25, something like that. So that's the difference. Okay, so that's, if anything at all, packing CHT is easy. It's just packed differently.
Yeah, let me add one other thing on top of that. I'm glad you mentioned that, Dan. So CHT or hydroxyapatite was first developed in 1952, I believe, 1952 by Tiselius. And it was crystalline material, which fractured very easily. And for a long time, people used that when they were thinking about packing CHT, which is much more robust. And they said, well, CHT is hard to pack. So I packed a couple of years ago, I packed a 1.8 meter column in Europe with CHT, and we were done in about 45 minutes. So it's very, very easy to pack if you know what you're doing. So please, again, please call us so we can help. You're also saving our jobs if you call with questions all the time, so please help us.
Yep. Okay, yes. Well, if it wasn't for the scientists, right, we wouldn't be here, so that's absolutely true. Okay, here's another one. Oh, all right, so this person, when I'm scaling up, are there any recommendations for bed height or compression factor, and additionally around flow rate? Who would like to take this one?
Well, I guess I will.
Okay.
It's this thing, right, where you wait a few uncomfortable seconds to see if anybody else is raising their hands. You don't want to wait. You don't want to go too fast, because then you take all of them. You don't want to wait too long because there's this uncomfortable silence. So I'm doing the best I can.
Yeah.
So
it's a good Goldilocks timing.
Right. Exactly. So we definitely have, or I have anyway, recommendations for bed height and compression factor. So in terms of bed height, you want to keep the contact time of your process stream the same when you scale up, and that's generally due to possible changes in things like, I mean, the most important thing is possible changes in viral clearances that you get if you change the contact time. So that's the thing that you want to keep the same. If you double the bed height, you'd want to double the flow rate, right? Having said that, people generally try to keep the same bed height and the same flow rate as they scale up. So people tend to scale out rather than up. The compression factor should basically stay the same, although if you're going from a very small to a very large column, there could be differences, and that's been studied by Stickle and Fotopoulos years ago, and we can certainly supply you with that paper. But by and large, the compression factor isn't going to change very much if you scale up from 5 centimeters up to a meter or whatever. So that's what I have to say.
Brilliant. Bill?
Yeah, and just as far as flow rate, at small scale, you're going to be able to go a lot faster because pressures are different. But as you get wider and wider, you're going to start running into pressure limitations, either primarily the hardware. I mean, most of the resins can handle, high pressure. But the flow rates, I remember a story, before I worked at Bio-Rad when Unosphere came out, it's like, hey, you can run 1,200 centimeters an hour. And I'm like, even us in the lab were like, well, but you can't transfer that to actual manufacturing. No pump can go that fast. So you just have to take those into consideration when you're doing your small scale development, that it's when you transfer to your pilot plant or your CMO, that they can actually continue with that same process.
Yep.
Brilliant.
Laura?
Oh, Dan, something to add. Go ahead.
Yeah, something to add here. So while we're on this packing topic, right? So I'm a product manager here at Bio-Rad, so I represent the marketing team. So I need to insert resources that we have on our website as well, right? That's part of my job here. And so we have packing videos for CHT and others, and they're very, very helpful. So in addition to the instruction manual for CHT perhaps, and all of our other manuals for the Nubia resins and all the polymeric resins, right? So they're in the manual, but we also have posted on our process chromatography website some packing videos that are really, really helpful.
Mm-hmm.
So,
and Laura narrates one of them.
Oh yes.
That's right. Okay.
Yes, that was a fun experience. Some really interesting terminology there that doesn't exactly roll off the tongue. Hopefully the B-roll from that, or the excerpts never make the light of day. Let's just say that.
We do have a blooper reel, Laura.
Yes. Concerned. Okay. All right, here we go. What are the scaling up recommendations for all of your mixed mode resins? And let me just push this so that you can read it yourself. Okay.
Well, I think we more or less just answered that. You typically want to scale out, not up. Bill is quite right. I mean, in theory, you could make a 100 centimeter tall column and run it 5 times as fast as a 20 centimeter column. But, as Bill said, pumps generally don't go that fast. And so it's kind of impractical. So out rather than up.
Yeah, and just, one thing to keep in mind with, say, A prime, it's a 50 micron bead. So smaller beads, your flow rates aren't going to be as high, as, say, a larger bead, or, standard ion exchangers, they're 85, 100, 120 micron beads. So you can flow those at faster flow rates. So those 50, but the reason you're using a 50 micron bead in a lot of cases is you want that resolution. So you're not going to necessarily be, screaming it through the column at that point.
Okay, and I just got a bit of clarification on that question. So these are recommendations that we're giving. Are they specific for CHT, or are they across all mixed mode resins, these scale up recommendations?
Yeah, they're across everything.
Yeah.
Okay. Thank you. All right. Let's get another one here. Okay. So, for acid sensitive monoclonal antibodies, what resin would be a good solution in the capture step from cell supernatant? And then, is buffer exchanging required? Who would like this one? Bill.
I mean, I think C prime could be a pretty decent option, as well as the A prime. C prime specifically because you're going to be running at a much more neutral or mildly acidic condition, you know, pH 5. And because the cell supernatant is usually going to be in that roughly 15 to 30 millisiemen, you shouldn't necessarily need to do any buffer exchanging because there is that salt tolerance with the Nuvia base bead.
Brilliant. Anyone want to add to that one? Bill got them all. That's good. Well done, Bill. Okay, coming at you fast and furious. Here we go. See if we can get through all of these in the last 10 minutes. So this person didn't get optimal selectivity from a mixed mode resin. Do you have recommended protocols for running a DOE?
Yes, we do. So you know, let me say a couple of things. So, you know, I did not get optimal selectivity. So quite honestly, then maybe that's not the resin for you, right? I mean, there's not going to be a resin that I've ever heard of that's always going to give you exactly what you're looking for. There are always trade-offs of one sort or another. If you mean you would like to improve the selectivity that you're already getting from a mixed mode resin, yeah, we have a number of publications on running DOE, and for A prime, 4A, and for C prime, those are generally using salt and pH as the two axes, right, of the DOE. For CHT, it's different. You're going to be looking at phosphate and salt, or phosphate and pH. But yes, we certainly have recommended protocols. And again, we have both 96 well plates and the robo columns and the spin column. So pretty much no matter what format you're able to use for your DOE, we can supply you with the things that you need.
Brilliant. All righty. I'm going to get us on to the next one here. Would you recommend CHT for messenger RNA purification? That's a good question. Bill?
Yeah, I would say yes. I mean, we have had customers that have used it. Typically, they'll use our type 2 CHT because it has the larger pores. But there are limitations. You know, because the pores are up to about 1,000 angstrom, you know, as the mRNA gets larger and larger, your binding capacity will be decreased. So it really would depend on, you know, how big of a molecule you're looking at.
I'll just add, it also depends on what you're trying to separate. You know, if you're trying to separate an N-length piece of RNA from N+1 or N-1, that's going to be pretty tough. Those kind of separations are typically done with 2 to 5 micron beads, and there's a reason for that, because the resolution is better. If you're looking for something that's a bit more general, yeah, CHT type 2 is a way to go. And although this is not a mixed mode resin, we have a resin called Nuvia HPQ, which has particularly large pores designed for large biomolecules, such as, for example, retroviruses, IgMs, and so on, that might be useful to try, which is it's just a simple anion exchange resin. So a bit off topic, but something to think about anyway.
Yeah. Good recommendations. All righty. Let's see if we can squeeze a couple of more in. Here we go. What are the recommendations for sanitizing a CHT column?
Good, Dan.
Dan.
All right, so I'll start this off, and then Mark and Bill, you can chime in. So first of all, it'll probably be, let's talk about a 3-step process. So after the run, you're gonna need to regenerate it first, right? And so for CHT, we recommend sodium phosphate at 400 millimolar to strip all the resins, retain the proteins off. After that, then the sanitization is NaOH up to anywhere from 1 to 2 normal NaOH concentration. Right. So to answer the question for sanitization is 1 to 2 normal NaOH, and then you would store the wetted CHT column at 0.1 normal NaOH. Okay. All in that room temperature.
Okay.
Yeah, let me add one. Let me add one thing to that, and that is again how CHT is different from other resins. Yes, it is at least in part a cation exchange resin, but unlike typical cation exchange resins, you don't want to strip with high salt. High salt actually makes some species like nucleic acids bind more tightly to the resin. So you don't want to use 400 millimolar phosphate plus 1 molar salt. You just want to use 400 millimolar phosphate. Again, all this stuff is in our applications guide, and please read that and even call us afterwards so that we can help you, because like I said, it's not like other resins.
Got it. There's Bill, go ahead.
Just one thing regarding the phosphate too. We have some customers that insist on using 500 millimolar sodium phosphate. Really, sodium phosphate solubility kind of peters out around 400. So if you need to go beyond that, you really should go to potassium phosphate, because that has a lot higher solubility. We do have some customers that strictly do potassium phosphate and potassium chloride even for their elutions. So depending on how much phosphate you need, just keep that in mind. And again, as Mark said, we can definitely steer you to the correct buffering systems for your application.
Brilliant. All right, let's get these last 2 questions. Okay. So are Bio-Rad mixed mode resins used in FDA approved products? Are we allowed to say? Who knows the answer?
Oh,
Dan.
I can start there. Yes, they are used in numerous approved processes around the world, right, worldwide. Our ion exchange resins and CHT and all our mixed mode resins. And so we have regulatory support files for all of our process resins to help you along to validate your process. And if you ever need any help, you just let us know. You contact us, and we can help you in any way.
Brilliant. Okay. And I think this will be the last one, unless one more sneaks in. Where can we get information about mixed mode resins? What are the best resources or best publications? Who would like this one? Bill.
I mean, specifically for Bio-Rad, definitely reach out to the Bio-Rad website. We have our process Chrome section there that has tons and tons of information. In addition, we have people like myself, Mark. There's actually a full team of field scientists like us globally that are available to come in and actually help you do work. I mean, obviously right now we're all kind of on lockdown, but under normal circumstances, Mark and I are traveling all over the US and the world. It's been kind of interesting being remote from home. Well, I work at home when I'm not traveling, but when I'm not traveling is the weird part this year. And then your local process reps would be a really good first point of contact, and then they will bring us in as needed to kind of help you guys develop your processes.
Brilliant. Yeah, I feel this this connectivity need from the three of you, right? Call us. I feel like you're missing out on that human connection here. So yeah, so please reach out to Mark and Bill and Dan if you do have questions. They really relish the interactions, I'm sure. All right, I think we've cleared through all of the questions. So well done. Excellent work, panelists, this morning. A round of applause to you all. And thank you so much for those questions, audience. I think that we managed not to, make anyone go too crazy or think too hard. So we definitely had the right people on the webinar today. All right, so before I sign off, it just remains for me to give a quick plug to some other coffee chats with experts in our series. If you would like to, you can go back and listen to some of our library of resources there with different topics such as infectious disease and qPCR, cell and gene therapy with digital PCR. And additionally, we have upcoming topics as well that you can sign up for around cell biology if you'd like to listen in live and have your questions answered. Okay, well, thank you once again to my panelists, and thank you all for listening. And that's all from us for now. So
Thank you, everyone.
I'm signing off. Bye-bye, and
Thanks,
thank
everyone.
you.
Stay safe.
Bye.